Okay, hello everyone. My name is Yannick Francioli, and today I'm going to present my poster. So my research focus on the processes that drive speciation at the whole genome scale and on the synergistic interactions of intra and interchromosomal coupling that produce genome-wide barrier to gene flow. So there's this fundamental goal of biology, that is to understand the mechanism that promotes reproductive isolation and that drives speciation, so the generation and maintenance of species. Theory predicts that speciation involves the aggregate effect of multitude of loci across the whole genome to produce strong genome-wide barriers that eventually lead to complete reproductive isolation and no gene flow at all between populations or species. Here we focus a lot on genomic coupling, which is this interaction between selection to keep the alleles of the same species together, and recombination, which inherently disrupt linkage. I won't go too much in detail about how we measure coupling. It's not the focus of this poster, and it's not the most interesting. But for the sake of this poster, coupling is a measure of intrachromosomal linkage. So what we did here is that we sampled 118 rattlesnakes from a hybrid zone in New Mexico between Crotalus viridis and Crotalus scutulatus. That's what you see here in figure 1. On the left, you have the parental, it's an admixture plot. On the left, you have the parental Crotalus viridis. On the right, you have the parental Crotalus scutulatus. In the middle, you have hybrids. And as you can see, we have a very nice gradient of hybrid indices. And what we did here is that we investigated the relation between coupling and other genome-wide inference related to selection and linkage, such as genomic Klein slope, which is indicative of selection, and interchromosomal linkage disequilibrium biased towards Crotalus scutulatus, meaning the LD between alleles that show resistance to introgression, scutulatus allele that shows resistance to introgression. What you can see here in figure 2 as the first result. So on the outer track here, you have Klein slope, which is indicative of selection. In the inner track, you have coupling, which is this measure of, as I said, of intrachromosomal linkage. And in the middle, we have the interchromosomal linkage. And what you see here in the panel on the right is really interesting. We found that variants that are in LD in this interchromosomal linkage also show higher Klein slope, so they are subjected to higher selection pressure. And also we see that there's elevated coupling coefficients, so there's also elevated intrachromosomal linkage in those regions, which already show this synergistic interaction of intra and interchromosomal coupling that produce a network, genome-wide barrier to gene flow in the hybrid zone here. We then investigated the genes that are in the region found to be in LD. So we found 324 genes in those regions. And we tested for gene enrichment and found that there's a significant enrichment for ion channel genes, which is really interesting because Crotalus scutulatus has a neurotoxin that specifically targets ion channel genes. So that suggests that there's a venom self-resistance driven incompatibility. The hybrids, they need to have the ion channel for scutulatus if they have the neurotoxin from scutulatus. What you see here on figure 4 is the network of genes that we found with their interactions, and all the colored genes are the ion channel genes. Finally, to further demonstrate this link between ion channel and the neurotoxin, we analyzed the linkage disequilibrium between the ion channel loci and the PLA2, the neurotoxin locus. That's what you see here on the top panel, and as you can see, the linkage disequilibrium is actually through the roof in the region that contain the ion channel genes. And there's also elevated coupling coefficients, so elevated intrachromosomal linkage. So there's a selection to keep those ion channels together in the hybrid zone as well. And so all of this really show a very nice example of genome-wide barrier to gene flow and how it is associated to key adaptive traits, and how all of this is further strengthened through coupling. And this is really the mechanism that is predicted to eventually lead to complete reproductive isolation and to speciation.